real world. It has obviously financial implications, but more importantly, it has implications for doing good work and having brilliant minds like yours and the faculty and everybody else who's produced things that really impact people and lives. And without commercialization and understanding the process and all the ins and the out, it just does ins and outs. It just doesn't happen. So hopefully all of you that are doing these presentations today and have worked so hard, I know over the last semester, appreciate the value of this. We certainly appreciate all the hard work you're doing and all the mentors that have worked with you. And I'm sure as you all see, the presentations will be absolutely fantastic. The judges, including myself and others, will lock ourselves in a room at the end and beat each other up and scream and throw darts at the board and spin the wheel and try to select how to distribute the $5,000. And of course, when all is said and done, it's there, right? The jaws, the absolute jaws of Shark Tank will be presented, and some very good group of presenters will be able to get their name on the plaque with the jaws. And of course, if you know anything about shark's jaws and shark's teeth, you understand those will be here when we're all long forgotten many years from now. So what a great way to live in perpetuity, in addition to the great work you're going to do by trying to help move these advances forward. So welcome to everybody. We look forward to a great day. I'm sure it's going to be a lot of fun. There'll be a lot of questions, I'm sure, coming from the scientific and the business side as well. And hopefully you all will engage with our presenters. So without further ado, Rob, it's all up to you.
Right. Thanks, Mike. So, yeah, let's get my deep voice on. Yeah, welcome everyone. And I don't know if you remember me saying to the class at some point, sharing the story of one of the students from the early years who asked me, Dr. Gordy, what do you do when you don't know what you're doing? Hopefully, you've now learned a little bit about this and know a little bit about what to do. You know, we know it's a course that it takes you on a course that many of you are not familiar with, but hopefully you've got a little bit more insight into the way that the translational machinery works. Okay, so part of my job here is to say thanks to people. So again, I'll start off by thanking you all. I know you've all worked very hard. You know, I know that sometimes this can be frustrating, but we're looking forward to, you know, your work product. I'm sure it'll be all of a very high standard. Again, we appreciate all the work you've done. I want to lead off by just thanking my administrative assistant, Linda. You know, honestly, I would not be able to do this without her. She is just a rock and anticipates everything and makes certain that the wheels grind. So thank you, Linda. It's really appreciated. I want to also say thanks to our sponsors. Ford Kemper from Woods Rogers. Unfortunately, Ford couldn't be with us today. He had been slated to be a judge, but he's closing on a very big deal. And so it was all hands on deck. We miss him, but we really appreciate the support he's put into this program. I'd like to thank Mike Friedlander. Mike has been stalwart over the years supporting this program, generously donating monies for both the prizes and also the reception afterwards. I'd like to welcome Harvey Brookins from the National Bank and Chelsea. Yes, Chelsea. I didn't get your Chelsea Grandfield. So Harvey and his bank also made a very generous donation to the prize pool. And again, we really appreciate the support. Another long-serving person on this venture is James Ramey of VTC Ventures. James is probably suffering from Stockholm syndrome. We've held him hostage for many years. So anyway, you got to see him during the course, and I always joke that he's a very effective teacher, and he's in the wrong profession, being a venture capitalist. He should have been a university professor. Okay, so let's thank a few other people. Ryan King, who was a former TBMH student and is now Chief of Staff of Roanoke College. Of course, Ryan participated in providing a lecture and his insights. Also Mason Wheeler. Mason's helped us out in lecturing to the class for the second year. Just a wonderful example of the TVMH program, and she'll be moving on shortly, postdoc. I'm not quite certain where. I think I do, but Mason, I'm sure, will update me shortly. Okay, so I also want to say thanks to all the mentors, and I'm just going to name everyone. So starting with the business mentors, Eric McNair. Thank you, Eric. Appreciate it as always. Marty Muscatello. I think Marty's online. Spencer Marsh. Cynthia Lawrence. You up the back there, Cynthia. Francis Farrell. Hey, Francis. Again, Cynthia, like Francis, have been long-term contributors to this program. Christy Wills. Christy, I think this is your first year as a mentor. And another first year mentor, Jessica Bertie from the veterinary school. And then we have our student IP mentors, Trista Yunro, Max Albrecht, Rowan Massinove, Alec Beck, Caleb Townsend, Sean Amani, and Eddie Regalski. So again, thanks so much for sharing your knowledge of this program with the first year students. We've just found this an invaluable part of the success of this program because of the idiosyncrasies of the information that has to be transferred from one year to the next. It's just very helpful to have former stepping up and helping out. Okay, and a couple more thank yous. Brad from the comms team. Thanks so much, Brad. As well as Leanne Kelly. Lena will be writing an article on this that should appear in BT News shortly. Also Clayton, you can see him at the back there, putting you in embarrassing situations with his camera. Also Matt Chittum, who prepared the flyer for the program. Okay, so hopefully, Mark. Okay, Mark. Yes, thank you, Mark. Yes, yeah, another long-term supporter. And thanks for making it down on a snowy day. I mean, goodness, you took your own life in your hands, Mark. So, okay, have I missed anyone else? Oh, yes, my friend Mark Van Dyke, former colleague from Biomedical Engineering. Mark can't be with us today. He has duties as Associate Dean of the Engineering School there at the University of Arizona in Tucson, but he's greatly missed. He used to be a colleague here at Virginia Tech, and every year gives an excellent lecture on regulatory aspects. Okay, so down to business. So you see the order. I'm just going to remind you that you have 7 minutes, and at the end of that 7 minutes, we're going to cut you off no matter where you're. All right. And then you have 5 minutes of Q&A. So I just want to wish everybody the best of luck. And yes, may the gift of the gab be with you. All right. Who's up first, Linda? Okay, so I'd like to welcome up Oncolak. Goodness. Thank you so much. Yes, certainly. Okay, you all ready? Correct. Yeah, good to go. Okay, so here are guys. 7 minutes starting now.
Hey everyone, we're the Oncolac team presenting a cancer screening medical device. Our team includes an exceptional biomedical engineer, Haley Levin, and 2 talented translational biologists, Kennedy Butler and Christy Vilkos, and then myself as a co-founder. This scene depicts the moment that I started my journey to create the Oncolac device. My mom was walking with a friend when she suddenly fell and stumbled. Further investigation led to her diagnosis of primary central nervous system lymphoma. And I remember seeing her brain scan for the first time. The tumor was the size of a golf ball. And I just remember wondering why we weren't able to catch it sooner. And that's what sought me to find better way to screen for cancers, because early detection methods are few and far between. So currently the problem millions of people face are delayed cancer diagnosis due to conventional methods that are often invasive, costly, and are not designed for early detection. Further, early detection is crucial, with survival rates significantly increasing if you can catch cancer early. For example, data from cancer.gov presented in 2022 showed that on average, patients diagnosed with a local or early stage, stages 1 or 2, had an 80% 5-year survival rate, while those diagnosed at a distal stage, stage 3 or 4, had a 22% 5-year survival rate. And a little as a 5% shift towards locally diagnosed cancers is estimated to save millions of lives in America alone. The solution to this is the cancer detection using OncoLAC device. It incorporates a graphene chip powered by a 3-volt battery and a voltmeter, and it all fits in a portable 6x3-inch design. The device works by adding a drop of urine to a reagent mixture, adding the solution mixture to the graphene chip, allowing 5 minutes for the reaction to occur, and then turning on the device to obtain a measurement, which can be used for a final concentration. The OncoLAC device utilizes a reduced graphene oxide biological field effect transistor to accurately screen for multiple types of cancers through a simple lactate urine test. Because of high physiological lactate levels greater than 10 millimolar are associated with nearly all cancers, it is the key biomarker OncoLAC measures. Lactate in the urine interacts with functionalized graphene surface, including redox mediators and enzymes, to produce a reduction reaction. The graphene chip allows for increased sensitivity to accurately measure lactate in solution and provide a real-time universal apparatus for cancer screening.
So here is the market opportunity for OncoLAC, and we based these projections on a few key assumptions, which are listed here. These stem from our projected sale prices, as well as PCP data from the United Health Foundation. We see a total addressable primary care market of about $6.7 billion over 10 years. From there, our serviceable available market, the clinics most likely to adopt a rapid, low-cost screening device, is $5.7 billion. The segment we can most realistically capture within our first decade, our SOM, is $1.15 billion. As you can see, this market is large, well-defined, and already motivated to adopt a rapid, low-cost screening tool to streamline preventative care. This chart shows exactly where OncoLAC fits into the current cancer screening landscape. Most existing devices are invasive, expensive, and are most effective at identifying disease in its later stages. OncoLAC fills a critical gap. It's low cost, easy to use, and operates at the earliest stages of cancer when screening has the greatest impact. Instead of replacing more complex diagnostic tools like imaging and biopsies, OncoLAC offers a new, accessible on-ramp to screening. It's meant to help clinicians decide whether or not further diagnostics are actually necessary. When we more directly compare OncoLAC to the current cancer screening and diagnostic options, its value becomes even clearer. Imaging is FDA approved and non-invasive, but requires trained personnel and is expensive. Blood-based testing is cheaper than imaging, but is still invasive and slow. Multi-cancer early detection tools like Gallery offer a more universal way of detecting cancer, but they are still expensive and not as accessible to most patients. OncoLAC combines the benefits of these approaches without the limitations. It's non-invasive, requires no trained personnel, costs very little per test, and is specifically designed for rapid, large-scale screening. It's because of this that OncoLAC is uniquely positioned to expand cancer screening to millions more patients, both globally and here in the US.
So here at OncoLAC, our business model is actually quite simple. We'll begin by applying for FDA approval as a Class 2 medical device. Our Phase 1 and Phase 2 and 3 clinical trials will be supported by grant applications and venture capital funds. By the year 2030, we'll begin to enter our primary target market, being the clinical diagnostic space.
So here's our year-by-year financial projection plan. Prior to pursuing venture capital, we aim to secure around $5 million in early grant support from the SBA, NSF, and ACS for research and development and phase 1 and 2 clinical trials. Because OncoLAC was developed here at Virginia Tech, our team believes that we would greatly benefit from participation in local accelerator programs such as the RBTC ramp here in Roanoke.
And as I said previously, our phase 2 and 3 trials be supported by venture capital funds. We will specifically be seeking $4 million in funding.
Around 2030, we will begin planning for an exit via acquisition by a larger biotech or medical device manufacturer such as Thermo Fisher Scientific, and this will help us with production and distribution.
And again, so we plan on generating revenue by the year 2030 by selling to our primary target market, that being the clinical research space. So think of your primary care physicians. Also, the secondary market being our preventative healthcare space. So think of your rural healthcare and community clinics. And finally, our tertiary market being the research labs, your biotech companies, and your pharmaceutical industries. And by the year 2034, at the 5-year mark, we projected to make a total of $5.65 million. And beyond that, we hope to shift our focus to the global healthcare market, reaching a total of $100 million projected revenue at our year 10.
Lastly, we would like to thank the consultants on our management team, one of them being Graphena, with whom we recently signed an NDA with for manufacturing parameters. We would also like to thank the judges for their time, and we will now open the floor for questions. Thank you.
Thank you.
Yeah, very, very nice, very clear and succinct. My question, as usual, coming from me, is about the science side, not the business side so much. So I want to make sure I understand your understanding of false negatives with this test, and particularly what the sensitivity is. You talk about many types of solid tumors that will respond to lactate signals, but how broadly applicable? What does it miss? And what are you going to do about false negatives?
Very good question. Thank you for asking that. So false negatives can be produced through extra lactate in the body, things like eating, working out, so fasting 12 hours before, and also not working out up to 8 hours in advance. These are things that we need to further look at in research and development. But as long as we can produce sort of a standardized testing frame and get out of most of the parameters such as the fasting and not working out, then we can produce less false positive results. And additionally, with the increased sensitivity of graphene, it allows us to test for a very, very small amount of lactate and then correlate that to larger volumes, such as physiological levels.
Oh, thanks. And just very quickly, I thought I heard you say towards the end there, your primary target is the clinical research space, and that surprised me. I thought your primary target was going to be clinical diagnostics. It's cool to be used
I
in
misspoke.
research.
It is the clinical diagnostic space. I'm glad you caught that.
Okay,
but yes,
you.
our primary target market would be your primary care physicians, your family internal medicine clinics. So you would go to your primary care office, your physician, they would recommend
No
the
worries.
OncoLAC
Yep.
test. Thank you that you caught that.
Looking at your target market, your total addressable market, why didn't you think about just going direct to patient, like Cologuard and some of these other tools?
That would be a potential future direction. After we've tapped into the market at the global health care scale, we definitely thought about leaning into the OTC or direct sales to the individual. However, we would have to seek out further FDA approval. It's under a 510K classification that will require more human factor testing and analysis to make sure it's at appropriate use for an individual at home. But that definitely is a potential avenue that we're definitely thinking about going into.
And if I could add on to that, kind of the vision that we had of when this would actually be in use is that a person would go into a clinic and maybe they're over the age of 50, or maybe they have family that has had cancer in the past, but they don't have symptoms of cancer at the moment. Tumors switch over to developing or producing excess lactate very early in the tumorigenesis process. So a clinician could hand this test to the person in-house and say, hey, take this. And it's a very early way to potentially catch something that could be attributed to cancer. And then the idea is they send them to further better diagnostic options.
Just real quick, one of the main advantages of your product is that it's cost, the low cost. And then when I looked at the projections, the financial projections, you only talked about the revenue in the top line, but you might want to think about look at the bottom line, the net income, because those investors are going to look at how fast is it going to take to get that return on investment, and not sure whether a $3,000 device could translate into a fast return on investment for the investors. It's not a question, it's more of a comment.
Online. I feel terrible, but I forgot to thank and introduce Kai Duffy. Kai is at the Corporate Research Center at Virginia Tech. Kai, hey. I just wanted to give you the opportunity to say hi and also ask any questions that you might have.
Thank you, Rob. I appreciate that. I'm sorry I couldn't be there in person today. Thank you guys for your presentation. I know how much hard work it is to do something like this. So can you talk a little bit about if your FDA timeline was extended, because I have never gone through this process, but I understand it takes some time. If it was extended by 2 to 3 years, how would that change your funding strategy and your runway?
Yeah, so I can talk a little bit about that. So we specifically chose a de novo classification, which actually does take a little bit longer than a regular 510K. I believe a 510K takes around 6 months, while the de novo would take around a year. It also does cost more. It's around 200K, while the 510K should be around 50K. So I think with extended time, it would just take us a little longer to get to market. But besides that, I think we plan for it anyways, just because the de novo should take longer than the 510K.
And we also plan on seeking out grant applications throughout the entire manufacturing process as we're seeking out FDA approval as well to support our ventures.
All right, thank you, Uncle Lack. And yes, and thanks for keeping the time.
Marty Muscatello put up a message. He asked to be promoted to panelist so he could ask a question. He's not being recognized as a panelist.
Oh, okay. Hey, Brad, can you promote Marty Muscatello to panelist? All right. Yeah, Brad's just taking care of something, guys. Just wait up a second. Okay, so here we have Remind Therapeutics. Nice job. Like the company name. And again, you've got 7 minutes, 5 minutes questions. And is Marty? Okay. Well, while Brad works, right. Hey, Marty, you've been promoted. You're now a panelist. So yeah, please feel free to ask a question in the next on the next presentation and subsequent ones. All right. So you've got 7 minutes. Good luck, and off you go.
Hello. Okay. Good afternoon, everyone. We are Remind Therapeutics, and our company's main mission is to sustain cognitive function for stroke survivors. Imagine that you survive a stroke, but you gradually start to lose your memory. Every year, many stroke survivors face a silent cognitive killer, neuroinflammation, which gradually causes cognitive decline. At Remind Therapeutics, we aim to prevent this.
One of the inspirations for our company was my grandfather, who passed away in 2022. He had an ischemic stroke, which resulted in severe post-stroke cognitive impairment, which eventually resulted in Alzheimer's, and eventually he passed away from a second ischemic stroke. And we ultimately want patients and their families to have an option, because right now, or when he passed, we had no options medicationally.
There are roughly 800,000 stroke cases in the US per year, in which 1 in 4 individuals is at risk for a second stroke. About 60% of stroke survivors experience post-stroke cognitive impairment, or PSCI, and only about 20% of the mild PSCI fully recover. Previous studies have shown that individuals with PSCI have increased pro-inflammatory markers, and that increased excessive long-term inflammation is associated with decreased cognitive function.
Without treatment, stroke patients are susceptible to detrimental cognitive impairment driven by this pathway shown here. Following the incident of a stroke, the SPAC pathway becomes overactive. This overactivation of the SPAC pathway disrupts the normal ion level in the brain, prompting an immune response that recruits these pro-inflammatory cytokines and reactive astrocytes, causing extensive damage to the brain and ultimately leading to post-stroke cognitive impairment. With ZT1A, we can prevent this. ZT1A acts to inhibit the SPAC pathway. And by administering ZT1A shortly after an incident, we can preserve as much of the brain as possible. And with monthly neuroprotective doses, we can halt the progression of any memory deficits and preserve the patient's quality of life. Treatment for stroke primarily aims to resolve the cause of the incident or treat post-stroke cognitive impairment. The current market lacks a catch-all therapy, but ZT1A brings us one step closer to bridging that gap. As you can see in this graphic, all therapies are capable of treating post-stroke cognitive impairment in some fashion, while tissue plasminogen activator, or TPA, has shown to decrease the risk of vascular dementia. That's heavily attributed to its ability to stop the ischemic event. This therapy does have its shortfalls. It does not reduce the risk of stroke recurrence, and it's only available within 4.5 hours of a stroke, which is about 3 to 8% of patients. Cholinesterase inhibitors are another group of treatment that's focused towards post-stroke cognitive impairment following the onset of symptoms. And while some have shown to improve cognitive function, the data is pretty inconsistent. Our product, ZT1A, is capable of preventing post-stroke cognitive impairment and reduces the risk of stroke recurrence.
As stated prior, 800,000 Americans suffer from a stroke per year. Of these, 185,000 are recurrent. Our company aims to treat over 100,000 patients who suffer from a stroke with ZT1A monthly. This route of administration will be through an injectable, which will be listed at a cost of $3,000 per dose. This is 40% lower than other comparable FDA-approved injectables. Currently, the global stroke management market is at $4.4 billion, but within the next decade, it is estimated to increase to $8.1 billion.
Our ZT1A therapy is currently in preclinical safety and efficacy trials, ultimately needing to undergo phases 1 through 3 clinical trials. Ultimately, this is going to require a lot of capital. And as a result, we've sought the funding sources on the left side of your screen, and as we're seeking additional investment in years 1 to 7 in order to cover our phase 1 clinical trial cost. Additionally, to cover our phase 2 and 3 costs, as well as to begin small scale manufacturing, which we would upscale during our phase 3, we're going to be seeking a 50% acquisition beginning in year 7, ultimately to cover this cost. Even if we don't receive an acquisition, our total roadmap has us generating revenue in 15 plus years and having us be debt-free by year 18. The way we're going to accomplish this is by capturing a very small portion of our total end goal of 100,000 monthly ZT1A users. In total, that's a $3.6 billion per year market if we were to hit 100% of it. And we did our calculations conservatively, starting at only a 1% market capture or SOM of that. Ultimately in sales year 1, with that doubling in sales year 2, and then doubling again in sales year 3. Ultimately, at that 1% level, which we believe we can exceed in the first sales year, we did our math off of that to be conservative. There's an additional potential market where once we are FDA approved, where we can use ZT1A in the ambulance immediately after TPA administration, and that has a potential market upside of additional 22 million per year. However, we would have to put it through the FDA pipeline again, but it would be much faster since ZT1A in the injectable form would already be through the FDA pipeline. To reach our break-even point, we are going to need a lot of capital, as I mentioned earlier. We're currently fundraising 22 million for our phase 1 clinical trial, and ultimately our total phases 1 through 3 costs should be around 107 million by our calculations. In our first 2 sales years at 1 and 2% respectively of the total market, that is just shy of $200 million, which would recoup the entire first year, 15 years of cost, even if we had to go into debt to do so. By sales year 3 at 4% of that market, that would be 264 million in revenue. Based on these calculations, we did a net present value calculation for our company at the acquisition point of year 7 to be $523 million. And that sale would be 261.5 million as a full return on investment for our seed stage investors. Our company would grow even further by the time we're making sales, which would result in 845 million of a total company valuation by the time we're making sales. So the reason you should invest in Remind Therapeutics is because ultimately we're on that path to a 50% sale where we're going to split 261.5 million equally between owners and investors. And at 4 investors at a $5.5 million input each, that would result in a $32.6 million windfall from the sale, which is a 492% return on investment. Additionally, we want our investors to keep 6.25% equity post-sale, which at the $845 million valuation comes out to be 52.8 million each in equity. So ultimately, we want to pay you back nearly fivefold at the 7-year acquisition point. And additionally, with no further input, your equity in our company would grow nearly tenfold. That's the reason why our investment in Remind Therapeutics is a good option.
With that, we'd like to thank Mr. Muscatello and Mr. Albrecht for their scientific and financial advising. Thank you very much, and we're open to questions.
So could you say a little bit more about mechanism of action? I thought I heard you say towards the end that you were going to move this into the field and administer it after TPA was administered. So TPA, if administered, as you note in your prospectus early on, is generally pretty good at clearing a clot from an occlusive. What sort of interaction? But more importantly, what is the actual mechanism of action, if you understand it, of zeta 1A?
So I think I have a slide here. So this is the kind of the pathway of the SPAC. And like we said, we mentioned we would use it with TPAs. TPA would get rid of the clot in the brain, but it doesn't solve the cognitive impairment completely. So the mechanism of action, we're kind of, the SPAC pathway is like overactivated in a stroke, and it's important because it controls these receptors here, notably the sodium-potassium cotransporter, which allows an influx of ions into the cell, and also downregulates this potassium cotransporter, which is controlling the flow of ions out of the cell. So we're having a lot of ions coming in the cell, not much moving out. So our mechanism of action is to prevent that upregulation and return to normal levels, where that sodium-potassium pump is not allowing ions in the cell, and the ions are allowed to move freely.
So what sort of off-target effects does that have, or what sort of data do you have in preclinical studies as to what that actually does in terms of even normal animals? What happens in a normal animal?
Our preclinical study in mice, let me get there a second. We have a lot of appendix slides. So this is a preclinical study in mice where they modeled stroke and administered ZT1A shortly after stroke and then for a few days following. And you can see ZT1A and its derivatives, most notably ZT1A, did show a 52% reduction of infarct volume in stroke. And then with the neurological testing, we see that it can significantly accelerate post-stroke function. And was there a second part to your question?
Well, what sort of off-target effects does it have?
Right.
Even in a normal animal, what happens?
So we're not anticipating a ton of off-target effects. GT1A is a non-ATP competitive binding site, so we don't expect off-target effects. It's something we would test in more clinical trials. But obviously, there are SPAC pathways are not only in the brain, so that would be probably an off-target effect that you would see. Yeah.
How would the drug be administered? Does it look like it was an IV?
It's an injectable, yeah.
Okay.
So an IV shortly after, so we have two phases shortly after a stroke, and with TPA is an infusion, so it would be administered with TPA, ideally, and then the neuroprotective is just an injectable.
Okay, great. I'm not sure how the patent works. So how long do you plan on the projections? You have a very long repayment period, year 7 and year 15. How long does patent typically last, and can generics come in and sort of take some of that market share?
So our patent is viable until 2043. Okay. So we have exclusive rights until that point.
Great, thank you.
She couldn't hear you. I'm sorry.
Kaya, did you have a comment or a question?
You guys have a patent currently on this product?
Yes.
Okay. And the funding round that you talked about, NSF, VIPC, and SBIR funding, are those currently under consideration, or have you received that funding?
So we've applied for that funding, but it has not yet been received. So in total, for my $22 million phase 1 total costs, it's factored in that that money is technically not in there yet.
Okay.
Question for you about your TAM. So you said you did like a top-down analysis and said the total TAM is 4.4 billion. I think that was like the spend on post-stroke treatments. Then you said your dose times your patients was 3.6 billion. So obviously that's a dramatic percentage of the total TAM. How do you think you're going to be able to justify that in terms of, getting insurance to pay for it, Medicaid, Medicare, et cetera?
Well,
Go ahead.
so ultimately, we are planning to sell our biologic at $3,000 per dose, which for comparable FDA biologics that are also injectables, they're traditionally sold at $5,000 per dose. And so ultimately, we're actually seeking a slightly lower price point, which would be advantageous to somewhere like a provider who's going to have to be fronting some of that bill ultimately when they're paying back.
Okay, well, thank you very much, Remind. Good job. As you guys know, I like the fact that you went for a drug, and we have that bonus hanging out there of $300 extra if you place. So yeah, so good going for an ambitious project. All right, so next up we have MitoCure. So we'll welcome them up on the up to the front here. All right, guys, you're all looking very smart. You ready to go? Okay. 7 minutes.
Hello, everyone. We are MitoCore, and we are revolutionizing cardiovascular therapies by creating protective mitochondrial-focused drugs that can improve the outcomes and save the lives of thousands of heart attack patients every year.
We all know someone who has suffered a heart attack and the life-altering consequences of these events. This is due to the massive amount of damage that occurs to a heart during a heart attack, which puts you at a lifelong increased risk of future heart disease and other health events. And while we've gotten a lot better in emergency medicine at treating the root cause of the heart attack, the blockage of the artery, this treatment has led to a paradoxical issue, reperfusion injury. You see, when we open up that artery again, this allows blood to rush back into that region and allows the hungry cells the oxygen to produce energy in their mitochondria. And they do this fast. However, this leads to the buildup of a lot of a side product, reactive oxygen species. It's so much the heart cannot handle it, and it creates an oxidative and inflammatory environment that results in 50% of the lasting tissue damage after a heart attack. So we are in desperate need of better protective measures for heart attack care.
And there's a big market for these preventative measures. We're looking at around 800,000 acute myocardial patients every year, 200,000 reoccurring, 600,000 new heart attacks per year. And so when we look at our addressable market, we're looking at that many patients in the US, as Aiden mentioned. And then for our obtainable market, we're currently looking at our first year of production. We're looking to reach 100,000 patients in the Southeast United States, mainly due to the high incidence rate of heart attacks in that area, specifically in the United States. And then by our fourth year of production, we are looking to reach all 800,000 patients in the US.
All right, now our solution for this treatment-based injury is our drug LL66, which is cardioprotective against ischemia reperfusion injury. Now, how exactly is it cardioprotective? LL66 is effectively able to reduce the amount of cell death from 70% to 30%, effectively protecting 40% of cells that would have otherwise died without our drug. Now, the exact mechanism of how LL66 does this, it's through its localization to the mitochondria. As mentioned before, cardiac cell death is largely caused by the damaging molecules known as reactive oxygen species, or ROS. The mitochondria serves to act as the source of the fire by actively producing these reactive oxygen species. Now, in ischemia reperfusion injury, a molecule known as histone deacetylase 1, or HDAC1, is key in the production of these reactive oxygen species. LL66 is effectively able to inhibit the action of HDAC1 and therefore prevent the production of these reactive oxygen species. Therefore, LL66 is effectively able to prevent the fire from even starting. An additional aspect of LL66 that makes it protective is its ability to work in a short time frame, allowing it to seamlessly integrate into current heart attack treatment. Now, with all of these great strengths of LL66, we believe it will meet the major unmet need of protecting cardiac tissue following a heart attack. Now, where exactly are we on our development timeline? So we have completed basic research showing great efficacy of LL66 in treating ischemia reperfusion injury in small animal models. Next, we'll be going into our IND enabling studies to further assess this and confirm our results into more cardiovascularly and metabolically similar animal models, and also to complete our chemistry, manufacturing, and control studies. Now, in tandem with our IND enabling studies, we will be completing patent extension to increase our exclusivity. And we'll be doing this through creating an alternative delivery system of LL66 to the heart through nanoparticles. And we aim to do this with commercial partnerships with nanoparticle companies. Now, upon completion of these IND enabling studies, we will then begin our clinical trials to first determine the safety and tolerability of LL66 in individuals, and secondly, to determine the efficacy of LL66 in treating ischemia reperfusion injury in patients. And we estimate that we will have FDA approval by 2034.
Looking at our financial timeline, we're currently looking to raise around $2.5 million in our seed funding round, as well as looking into SBIR and AHA grants for that at a market value around $15 million. After these IND enabling studies, we're looking to raise $20 million in our Series A funding at a market value of around $80 million. Looking at our 2A and 2B clinical trials, we're looking to raise $150 million at a $400 million market value. At this time, we're looking at pursuing acquisition. However, if that does not occur, we're looking to raise $300 million in our Series C funding at a market value of around $5 billion. And looking to raise this type of money, we're looking at large biomedical venture capitalist funds.
And this brings us to our business model. In our first year, we plan to target major and regional hospitals in the Southeast United States, as this has the highest incidence of myocardial infarction. Continuing, we will ensure that LL66 has value-based pricing, meaning that avoiding rehospitalizations from heart failure will actually save significant money for the hospitals. This will be critical for passing our drug through the pharmacy and therapeutics committees, as well as the value analysis committees, and getting our drugs onto the formularies quickly. Continuing with these successes and with the positive connections we've made with expert doctors in the field, we will continue to expand until LL66 is used to treat myocardial infarction in every hospital in the United States.
Cardiovascular and mitochondria therapeutics are both highly acquisitive sectors. Since 2019, large pharma has completed 10 plus cardiovascular M&A and licensing deals. Importantly, mitochondria space is also very active with acquisition totaling 6 plus mitochondria targeted deal in recent years. So LX66 is the only therapy offering a mitochondria localized class 1 HTAG inhibitor with direct rapid protection during the perfusion, giving it a first in a class profile and a clear competitive edges. So current IR injury treatment lack mitochondria specificity and the only partial or indirect protection.
What do clinicians think of our drug? Well, they think it is revolutionary. They are very knowledgeable about the need for better protective measures and how these heart attacks cause drastic complications in their clinical setting.
Finally, this is our management team. Our team consists of many people that are proficient in cardiac IR injury biology, as well as translational skills. We brought on experts and consultants in the field with experience in creating startups, translational methods, and interventional cardiology.
Thank you, and we'll take any questions at this time.
What are the potential off-target effects of the drug?
Yes, so histone deacetylases, or HDACs, are very important in maintaining cellular homeostasis. With ours specifically being in the mitochondria, they're very important in maintaining mitochondrial homeostasis, such as assisting in repairing mitochondrial DNA and assisting with maintaining energy balance. So if there is repeated dosing of LL66, we'd anticipate there to be potential mitochondrial dysfunction, but the great strength of LL66 is it's being administered acutely at one time before the reperfusion event, so there isn't going to be those potential toxicities. But we would also, in our IND enabling studies, have a great emphasis in our formulation to help mitigate LL66 being in long-term circulation.
Marty, do you want to go ahead?
Yes, just a question about the return on investment. If I'm an investor at the early stages, I'm not sure I quite followed the math, but if I invested, let's say, $100,000 early on, what kind of return would I expect by the time we exited or went through an exit? Can you kind of walk me through that a little bit?
So we intend to have made our money back and be profitable by the end of year one if we were able to sell 100,000 doses of our drug. And so I believe if we go to the other numbers, it should be a several 100% increase in your profits.
And those would start on the what's year one? When you said year one, how many years is that to get to year one for profitability? Was that about 15 years maybe or something?
So our estimate of FDA approval is around 2034, so about
Okay.
9 to 10 years.
Thank you.
You have a large, big ass or big fundraising. Why the big costs on the fundraising? Is it because to prove the efficacy of the surviving cells, or why does it seem to be so high?
Yeah, well, it's really large because, well, one, it's a cardiac drug. For one, it's also administered acutely. You can't plan a heart attack. Excuse me. And so that's a big reason for it. Sorry. Looking at the average value of how long it takes to make a medication, 800 million to around a billion, are the common numbers that we get. Those numbers are a little bit lower right now, but this is a very conservative estimate. But just due to our drug, there's really no drug that has been shown to consistently do what ours does. And so we do anticipate longer trial times.
What do you know about the time between the administration of your drug and reperfusion and the efficacy? How much does it matter? How tight does that window have to be? Do you have data on different effects with different times?
So from our preclinical studies, we have seen that we administer LL66 approximately an hour before. So the effective time frame is an hour. So we'd like LL66 to be emitted directly before the reperfusion event. So usually this will be done in a cath lab or an emergency department scenario. So this will be done in the preparatory stages of like administering an IV. So it would be done right immediately before that.
But you have data at different time points.
As of right now, we have only showing it in that initial time point of doing it within that 60-minute
1 hour.
period. Yeah, so we would have to robustly see at exactly what time points are the most effective for LL66 to be.
Yeah, it might be worth investigating that. Okay.
We do have it in mice, which do have, I mean, their metabolism is much faster. And so that's why we want those ID database studies, say in pigs. Because an hour for mice, I believe, is around 6 hours in humans. I believe that's what the study said. I could be wrong. But that is a really fast metabolism, is why it's so important to really, really check how it works with more, with models that are closer to us.
Can you go back to your development timeline? The slide where you had the, yeah, there you go. So why do you have nanoparticles loaded with L66 after your IND enabling studies?
So we're actually going to be doing it in tandem with the IND enabling studies. So we'll be doing our original formulation of the IV administration and also that of doing the nanoparticles, because we want to use this nanoparticle solution to do inhalation rather than IV, because it allows more rapid delivery of LL66 to the heart. So by doing it within the IND enabling studies, we don't have to repeat anything and we can continuously go forward.
Another drug, yay. Okay, I'd like to welcome up to the front Train X Exabio. Is that the pronunciation? Okay.
Good afternoon, everyone. Today, we are thrilled to present you Trenexa Bio, where we are transforming hemorrhage management in emergency and trauma care settings. Over the next few minutes, we'll walk you through what our mission is, what our product is, and how we are uniquely positioned to capture this market segment. And this brings us to our flagship product, TXA Guard, which is a handheld device that stops severe bleeding at the source by delivering medicine directly to the wound. And the opportunity, TXA Guard will help us target a market worth $3.5 billion by 2035 with patent protection extending to 2041, helping us to achieve our mission, saving lives before emergency care arrives.
Okay, so what is hemorrhaging? So hemorrhaging occurs when blood ruptures from your vessels. And so our device aims to answer the question of how can we treat hemorrhaging more efficiently? And so it's important to understand the impact of hemorrhaging in the US. So there are around 3 million people that are diagnosed with some sort of bleeding disorder. There are over 300,000 emergency department visits for anticoagulant related bleeding yearly. There's around 450,000 visits for nosebleeds, and it's about 60,000 deaths per year due to hemorrhaging, and around 31,000 of those are preventable. And then roughly 2% of the US population has some sort of chronic non-healing wound, and emergency departments across the country treat around 8 million people every year for some sort of laceration. And so who is our market? We plan to market to outpatient wound care, oncology, hematology, ear, nose, and throat doctors, EMTs in emergency departments, as well as orthopedic surgeons. And that puts our market at around $3.5 billion.
So our product is designed to be a very elegant and simple and can be operated in very 3 seamless steps. First, you press and activate the device, which releases a metered topical solution onto the sterile applicator pad. Then the user presses this applicator pad against the wound. And the third step where the magic happens, TXA stabilizes the clot. And we also have an option for our users for an additional adhesive backing for hands-free compression. And so product encompasses 3 interconnected innovation. Our proprietary TXA formulation, which is engineered for rapid absorption and clot stabilization. Our device innovation, which is coupled with a frangible reservoir with a pressure distributing convex plate for optimal wound contact. And our targeted TXA delivery, so we can circumvent the harmful systemic effects of TXA. And so this is because we have a multi-layered patent protection on all of our 3 innovations. Our competitors can't copy us, and we'll have an extended 20+ years of market exclusivity for our commercialization.
Our business model consists of 3 different phases. Our first phase will focus on getting FDA approval through clinical trials, and this will be for both human and animal use. And then after FDA approval, we will implement our product throughout the Blacksburg and Roanoke areas in Fralin hospitals, emergency centers, as well as to EMTs. And then within a year of FDA approval, we will expand to hospitals throughout Virginia, as well as neighboring states such as North Carolina. For phase 2, this will be within 3 years of FDA approval, and it will be an expansion across the East Coast, as well as implementing our product for military usage for emergency situations, as well as in animal hospitals. And then for phase 3, this will be within 5 years of FDA approval, and we will push for a nationwide expansion of our product, as well as getting insurance coverage so that patients with bleeding disorders can have this product at home for emergency care.
And so how do we stack up against the competitors? So TXA is currently used for oral and IV formulations, but once FDA approved, our product will be the only topical TXA on the market. We plan to reduce hospital admissions. It has applications for various fields of medicine, as previously discussed. Since it is a one-handed device, it's easy to use, and compared to our competitors, there's low toxicity.
So straight to the financials, because this is a small business, we do not expect to make large profits in the first few years. Actually, our first 3-4 years is going to cover our clinical trials and FDA approval, after which we expect to break even by the 4th year and eventually climb up the profit ladder. We expect about $10 million from grants, of which we have already secured $3 million from the Bleeding Disorder Foundation. And so today we are here asking for $5 million to start this great business, of which will give you 20% of our business, which is now valued at $25 million.
So our management team will be consisted of the 4 of us as well as 3 consultants. The 4 of us will have different areas of expertise such as public health and clinical implementation, as well as marketing, scientific discovery, sales and financial modeling, and future expansion and product development. And then we also have 3 consultants that also have different areas of expertise, where one will be helping us with the product itself, another for any financial concerns, as well as a clinical consult. And then thank you for your time, and we'll now open the floor for questions.
Kaya, I'll give you the chance to ask the first question if you have one.
All right, let me see which one I'd like to go with. Thank you for your presentation, everyone. So I think you mentioned a $6 billion hemorrhage control market as part of your presentation, if I'm remembering reading through this correctly. What portion of that is addressable for a topical device like you're suggesting?
Okay, so these are the market options we have. Around 54.2% of the hemorrhage control system market is for hemostatic devices. And so we plan to enter into that sector, if that answers your question.
Yeah, thank you.
Mike?
So as I understand it, your device has a pressure component as well. So I'm just curious, first of all, about controls and what just the pressure did and what the differential is with your compound on the wound.
Yeah, so initially the user would have to press it against this wound. And so TXA has been around the medical industry for about a decade, and it works within minutes. And so the only problem with the IV and the oral routes has been the side effects of it, because we are applying it topically and locally over the wound. That solves our problem, and the hemostatic pressure is enough to be applied by a user and to be able to sustain it for a few minutes. That's what we are anticipating.
Okay. And at the beginning, you showed all the people on anticoagulants and all the regular lacerations, aging, etc. What sort of differential responses do you get for those different conditions with hemorrhage?
So right now we are placing a product for severe to mild surface bleeds. Think of epistaxis, lacerations, but not for internal cavity or perhaps even to dental extractions. And that's all, not extending it to a surgical suites. So within those realms, the operating period would be around 10 to 15 minutes, and that's all.
Yeah, I was just wondering if you have differential results under the different conditions that you're using to treat this.
So there are animal studies as well, but no, specifically not for the different things.
Okay.
I'll also add topical TXA is currently used in surgery for off-label, so it's just not FDA approved. So surgeons do use it for orthopedic surgery, and they use it for tooth extractions and stuff. So it has applications.
Yeah, thank you. I might have missed it. So how much does this cost for the end user to use?
Okay, so each unit will cost $260.
And does the end user have to go through any training in order to use the device?
So it comes with a little manual that you read, and then you can easily apply.
So it's sort of like an EpiPen for hemorrhage?
Yes, exactly.
Okay, thank you.
I had one. In your development plan, you said in year 5 is when you would try to get reimbursement via insurance. Why wait until year 5?
So the insurance was more focused on the patients that would have this at home, but we would still like allow the patients to try to get insurance coverage if they were using this like in a hospital setting. But that was more focused on just getting patients to be able to have it at home as like a prescription that their doctor would prescribe, and then they could get it covered by insurance.
Okay, thank you.
Yep.
All right. Thank you, team. Okay, you got 2 rounds of applause. That must be good. Right, so we're now welcoming up Aurelia. Okay, guys, you've got 7 minutes.
Good afternoon, everyone. We're Aurelia. We are a company seeking to transform care for postpartum hemorrhage management. My name is Lucas. I'm here today with Shubangi, Reese, and Harika, and I would like to introduce you all to Maria's story. She's a 28-year-old woman, 9 months pregnant. She just gave birth to a healthy little boy, but 30 minutes late, she starts feeling dizzy, and that's because she's losing blood, but not just regular amount of blood, but a lot of blood. At this point, she already lost more than 500 milliliters of blood, which is considered a case of postpartum hemorrhage. Drugs will not kick in time to help her. The tamponade balloon cannot handle this amount of blood. So the doctors decide to proceed with bimanual compression, which consists of introducing their face into her vagina and compress both inside and outside and compress her uterus to stop bleeding. As you might think, it's very intrusive and very painful, and in her case, could not stop the bleeding. At this point, her husband is staring at her from outside of the room, holding their baby in his arms, crying because he's gonna lose his wife. This is a reality for a lot of people. 12% of maternal deaths are due to postpartum hemorrhage in the United States. Also, 165 people die daily from postpartum hemorrhage, and all this causes an economic burden of $1.8 billion per year. But we can save her. And to do that, we develop a low-cost, accessible, and easy-to-use postpartum hemorrhage management device to reduce maternal morbidity rates on a global scale. So there is hope, and we call it PPH Butterfly.
So when Maria started bleeding, her team employed BMC or bimanual compression, but inserting the fist up the vagina and sustaining that amount of pressure that's required to control the bleeding or stop the bleeding, it was not feasible. And after a while, we noticed that her bleeding started to increase again. And this is where the PPH Butterfly came in, which is our solution. After multiple rounds of patient feedback, multiple iterations of the device, as well as clinical studies, we developed this device that is easy to insert as well as reduces clinician fatigue. So when inserted, when folded, it slips into the birth canal where it opens up against the uterus and applies pressure against the uterus. Now, all the physician has to do is apply pressure externally on the abdomen. In this case, the uterus is constricted between this external hand and internal platform, and this constriction allows the uterus to start contracting, and it helps reduce the blood flow. And once the bleeding has stopped, this device can be folded back in, and then it slips out very safely and atraumatically. So in a moment where every single second matters, the PPH Butterfly is a pretty useful device. It provides stability and strength to the physicians and also helps patients like Maria be saved. And our goal with the PPH Butterfly is to use it as a first line of treatment, even before any uterotonics or other devices are given to the patient. And the PPH Butterfly will basically stop the blood flow pretty in the early stages, and it helps reduce the flow of blood pretty early.
Current standards of care for postpartum hemorrhage include uterotonics. These are pharmaceuticals such as oxytocin and TXA, but these interventions are quite costly and they're not always accessible in rural environments, and there's a delayed response in their timing to work to stop hemorrhage. So the JADA system and Bacri balloons are the current medical devices that are implemented in hospitals for postpartum hemorrhage.
yet these devices are very costly and not applicable to cases of severe postpartum hemorrhage. And then we have bimanual compression, which is both invasive, painful for the mother, and extremely tiring towards physicians. As you can see in our market, the PPH Butterfly is the only both cost-effective medical device that is available in all rural areas and that also reduces physician fatigue, unlike our competitors on the market. For our implementation plan, our FDA 510K established the PPH Butterfly as a Class 2 medical device. The NCATS grant helped us roll out clinical trials in local hospitals in the area, and our Continuation of Excellence grant helped us roll out the PPH Butterfly at both the state and national level. For phase 1, we had our clinical trials in the county. In phase 2, we have the Virginia Hospital Adoption Strategy. In phase 3, we have U.S. distribution to both rural and urban hospital systems. And phase 4 consists of globalization of the PPH Butterfly.
Looking at when we break even, we expect to break even in 2028 with a little over $2 million in our cash balance. Our manufacturing cost is $1.45, and we expect to sell the device for $39, which show a profit margin of 98%. Looking at our acquisition potential, are very much specialized in maternal fetal medicine with a focus on women's health with surgery devices and as well as a partnership with UNICEF.
So postpartum hemorrhage is not far away from our reality. This is Dr. Witham from Fralin Clinic. She says that it's a high clinical interest area in their department. Their rates of PPH are twice national average at 6%, and they would significantly benefit from additional management strategies, considering they're currently using the bacteria balloon and the JADA. And just to give you all an update on Maria's case, I mentioned before, we can save her life, and in fact, we did. She joined our clinical trials for our product, and she said it's much less intrusive, it wasn't painful at all to her, and stopped the bleeding. She says she's relieved the butterfly device worked for her. And as Aurelia, we're seeking to transform care and save lives. And for that, we are asking for $600,000 for funding nationwide hospital adoption strategy. This is our management team. I would like to thank you all for your time and your consideration. We're open for questions. Thank you.
Yeah, you asked for the 600,000, but are there any substantial raises that have to occur after that?
That's a great question. So right now, do you mind giving me that? So right now, our total addressable market ranges from 150 to 600 million, but right now is at 300 million because we're still focusing on the nationwide, which is why we're asking for 600,000, because then we can expand globally, because right now we are still focused in Virginia and the United States. We want to expand the global partnerships.
Yeah, well, once you do that, we'll then have to raise more in order to, expand the market.
Yeah, and we plan to, that will probably expand to $1 to $2 million.
Okay, because I saw on your financials, you had deficits, negative cash positions, and then your break even. That's probably want to add that to your 600,000 because you're going to need that to continue to operate. Yes.
Have you considered, or does this involve the use of haptics for the physician to get any feedback on the pressure as they're delivering the device, or is it completely doesn't have anything like that? Is that right?
So the device can sustain about 1 Newton pressure, which is about 10 kilograms. But the amount of pressure that we need to apply is kind of different for each patient. So, but the PPH Butterfly can sustain.
But in terms of the physician knowing the amount of pressure without haptics or something, they're just feeling if it gives or buckles.
Yeah, so in order to control for that, they basically, the length between the platform as well as the, let's see where the,
You can explain it without showing the picture. It's okay.
The length between the platform that we can see as well as the handles is about the length of a medium cusco speculum, and that kind of helps us adjust for the pressure and even the
And are
physicians.
these one-time use disposable or reusable, sterilizable?
So for now, they're one-time disposable, but we are hoping that in the future we are able to make them.
I can add to that. We are currently working with the University of Liverpool and the actual inventors of this patent to develop a reusable component to use in the future.
Okay. And why is your margin so high? Other than, I guess that's a good thing to make a lot of money, but why is it 98 or whatever it was?
Our margin is quite high because this is an area that always needs more help. There's a huge global need for PPH treatment so that we can help reduce mortality rates in maternal health worldwide. So that's why it's such a big margin, because it's an ever-growing market. There will always be more births happening. There will always be production of this device to assist in that to reduce the death rates.
What's the cost differential between this and the balloon, which I assume is probably the standard of care?
So the difference between this and the cost differential of the balloon, so the balloon is about $600 per single unit, and our device is roughly $39. So we are 75% cheaper than our alternatives, which is crucial in spreading our device to rural areas and countries that are not as well developed.
Questions online?
Thank you, Dr. Gordy. My question was about cost of goods and cost to produce. So it's been answered already. Thank you.
Thank you, Kai.
Who do you think would be the most likely acquirer for this? And do you know what other companies similar to this have been acquired for?
So the most likely company to acquire for this would be Johnson & Johnson and our partners through UNICEF, and then any other local hospitals that agree to pick up on the patent. So for example, we've worked with Fralin for our phase 2 clinical trials.
Sorry, I mean acquire the company, not be a customer.
Johnson & Johnson.
Okay. If there are no more questions, well done. And thank you. All right. Apparently, I can't be trusted with a microphone. So next, I'd like to welcome up Ovi. Have I pronounced the company name? Okay, good. All right. So welcome, Ovi. And are you ready, Linda? Okay, you've got 7 minutes. And there's your microphones and the slide advancer.
I wish. 2 simple words, but in cancer, they mean the whole universe. It always comes from a place of regret, gratitude, and the painful awareness of how precious time is. I was 14 when I first visited a family friend that was diagnosed with stage 4 ovarian cancer. I remember entering her house feeling the quietness and heaviness in the air, as if like even her home knew it was her last day. And it felt like, and I saw her kids sitting next to her, not knowing what the future holds for their future mother, their whole world. I thought Sarah was going to be angry, but instead she said, "I'm not angry. I just wish I had more time. I wish I had known this earlier." This is the reality of ovarian cancer, unfortunately. It's the #1 deadliest gynecological cancer in the US and worldwide. And that's because 70 to 80% of ovarian cancer cases are diagnosed at late stages. And that's because symptoms are vague and easily mistaken. And the most importantly, there's no standard screening test currently that exists to help with early detection. And that causes over 20,000 cases and over 12,000 deaths expected in 2025. That's 250 women dying every week. And it costs the US $6.4 billion per year. This is a simple graph showing you the comparison of ovarian cancer death numbers compared to other gynecological disorders. Cancer. And as we can see here, it truly is an orphan disease, is the deadliest, but however, it has minimal studies currently going on detection, on early detection, unfortunately. So this makes you wonder then, why is it so important to detect cancer in its early stages? Simple as it is, the sooner you find out, the better and faster you can treat it. And that can raise 5-year survival to 94%, according to American Cancer Society.
Like ovarian cancer is met with no screening methods. We're letting Sarah down, we're letting patients like her down and their families down. So where can we start? 2 things, sensitivity in screening and accessibility of screening. And for that reason, we bring you Ovi, our microfluidic biochip. Ovi functions very simply. With only a finger prick's worth of blood, and within seconds, our device is able to detect at very early stages ovarian cancer. The way it works is by having blood travel through these microchannels, and at specific microfluidic serpentine sites, as you see here, exist a wide array of combination of different antibodies. Now, these antibodies specifically bind to unique markers of the ovarian cancer cells. And when this happens, an electrical change is detected by our device. Now, currently, the CA125 biomarker is the traditional biomarker used to detect ovarian cancer. But as you can see, the sensitivity is not reliable, which is why there is no current screening method. Some attempts have been made to use a combinatory approach to these antibodies by implementing HE4, but that's still too low. So in order to address this key factor of sensitivity, Ovi will implement the combination of APOA1 and TTR with CA125 to result in a 94% sensitivity at stages 1 and 2. And the way this system works is simple. You simply add the Ovi cartridge onto a reader. This reader transmits information detected by the cartridge to either 1, a computer in the clinic by the physician, or 2, in the patient's home. Using Wi-Fi capabilities, it can connect to a patient's phone via an app. And this device is officially licensed by the New Jersey Institute of Technology by the inventors, Dr. Lee and Dr. Nuna.
So our primary target customers will be the healthcare systems, such as the hospitals, because they have a large demand of early cancer detection tests. The OB/GYN clinics are also our target customers, as they have a similar demand as the healthcare systems do, and they can perform the test for patients who come visit them with vague symptoms. And the patients can also get tests at home using our device if they're experiencing vague symptoms with family histories or having a high risk of developing ovarian cancer. And here is a cost breakdown of our device. We offer a starter kit at $100 per kit, and additional cartridges can be purchased at $25 per cartridge. And here is a chart of our pricing strategy. The gross margin profit of our device will be 38.8%. And why Ovi is superior? First, it's because the cost per test of our device is much cheaper compared to our competitors. And our device is able to deliver the test results within 1 minute. The sensitivity and specificity of our device are among the top level compared to our competitors. And at the same time, our device achieves a much better balance between these two parameters compared to our competitors, with both of them are above 90%. Additionally, our device is labor-free, which means it doesn't require additional sample preparation, lab tests, or device maintenance. It allowed the patients to get tests at home, and its non-invasive nature allowed the pregnant women to get tests as well.
So beyond ovarian cancer, our secondary market lies in 2 of the most common gynecological cancers. One is cervical cancer and the other is endometrial cancer. Now to bring Ovi to the market, we anticipate raising $8 million from investor capital, and we also anticipate sourcing other additional funding sources from these grants. Now, if you look at the chart here, both in our base case and upside case, Ovi turns from negative cash flow into profitability, where we break even at year 3. Now, let me walk you through our 5-year implementation plan. In 2026, we focus heavily on analytical validation, where we generate the data needed for regulatory approval. 2027 moves us into clinical readiness, mainly with FDA submission. 2028 is our inflection point, where we scale up to generate 1 million kits, capturing 10% of the ovarian cancer market. In 2029, we accelerate further, producing 3 million kits and 500,000 cartridges, generating a profit of $158 million. And also we would be extending our development of the cervical and endometrial cartridges. By 2030, we anticipate raising a revenue of $1.1 billion, and our profit would reach $950 million. At the end of this 5-year cycle, we would be returning 20% back to our investors, which is more than $190 million, and we would be allocating 2% royalty per cartridge sold, so that our inventors are rewarded proportionally as Ovi scales to millions of annual tests.
Upon speaking with numerous gynecological oncologists, it is clear that the common theme is to address factors of sensitivity and accessibility. Dr. Osborne here at Caribbean Clinic attests that Ovi could be a device to address these things. Thank you.
Questions?
Hey, nice job. Is there anything known about a pulsatile nature of release of the antigens from these ovarian tumors and or a circadian rhythmicity that would suggest that there are certain times, either in the course of the disease or a course of a day, that the test would be more likely to detect an early stage cancer?
May I ask the pronunciation of the first word? Volatile, did you say?
Pulsatile. release,
Pulsatile?
versus a continuous level. Are there, earlier on it moves to stage 2, et cetera, levels go up, go down, et
Certainly.
cetera?
Yeah, so actually one of the biggest issues with the current screening methods are CA-125, which rises and decreases along many different factors like the menstrual cycle, for example. Biomarkers like TTR and APOA1, which we use, are actually expressed at a, their profiles are expressed at a lower level opposed to a higher level, allowing us the increased sensitivity. So it would account for any circadian rhythmic factors, like you mentioned, present in CA-125 at the moment.
Okay, thanks.
I'm not sure if there's any other like cancer detecting devices that person can use at home. But have you thought about sort of the psychological factors that allowing people to find out that they have cancer at their house instead of around professionals?
Yeah, so that's a really good question. We spoke to doctors and patients, and they were saying that they would prefer knowing earlier than later. And so even a lot of patients go in with vague symptoms, and which is why ovarian cancer can progress faster within 1 to 6 months to stage 4. So that doctors they want to do this, they want to implement this to be able to address those fake symptoms. And since it takes only 1 minute, they can just do that test, and then from there they can move forward to the other test to confirm whether it's ovarian cancer. It definitely causes stress, but as Dr. Osmore said, it's way better than the stress of knowing later that you have ovarian cancer, because it's the deadliest.
Can you walk us through your total addressable market?
And we wanted to tap into 10% of that market because the ovarian cancer screening market we estimated that it's 10 millions. And
10 million patients?
10 million screening tests.
10 million tests, okay.
Like
so,
the
so which
CA-125
patients would that, is that somebody who suspects it or just all women over a certain age or?
No, the first, the screening test would be firstly the CA-125 ELISA test, and that's where we based our marketing, and it's supposed to be at like it's estimated at 10 millions, and we wanted to tap into 10% of that market. And so that's why we started with producing 1 million starter kits and scaled up from there.
Did you want to add something?
Yeah, I wanted to add that this test would be recommended by doctors and the patient themselves to do if they have it in their history or if they have the vague symptoms. As soon as they go in, since ovarian cancer is the deadliest, the doctor recommends doing that test right there as they're meeting with the patient. And if it gives them a high levels, and it's most likely ovarian cancer, they move forward. So it's people with vague symptoms, also in their history, and as well, every woman is recommended to do this test.
Kaya, any thoughts or questions?
I think this is connected to the question that was just asked. I was looking from 500 units in year 2 to 1 million kits sold in year 3. That feels like a very large gap to cross. Can you talk a little bit about how realistic you think that is?
$500 million. We started with 1 million starter kits.
Yeah, you said 500 patient multi-center pilot in year 2, going to a launch of 1 million starter kits in year 3. So from 500 to 1 million.
Oh, well, in the previous one, we were talking about a study. The pilot study would recruit 500 patients,
Okay.
because it made logical that the data from the 500 patients would give us good data to move towards manufacturing. And in the first year, as I said before, that we wanted to tap into 10% of the 10 million market. And that's why we chose the number that we want to start with 1 million starter kits.
Okay, okay. And do you have in here, and I just maybe missed it, I apologize, your cost of goods for producing?
Oh, let him answer. Answer. You don't need a slide, just tell us a number.
The price to produce the reader will be $30 and the price to produce the cartridge will be $15.
Okay, thank you.
Great. Good afternoon, everybody. We are Photovis, and we are bringing new light to your vision. So just to start out, I wanted to introduce our team. My name is Sam. I am your CEO. This is Jade, our CSO, Emily, our CFO, and Shabman, our COO.
All right, so I want you to imagine for a second you're a patient with diabetes. Your days are already hectic just trying to keep yourself alive, so it's only natural that things fall to the wayside. You've had 20/20 vision your whole life, so you never really understood the purpose of an annual eye exam until one day you start to get these weird black squiggly lines in your vision. You try to ignore it, but as time goes on, it gets harder to go to work, to drive, to see your spouse, to see your kids, to see your grandkids. And once you finally get to the eye doctor, they let you know you have late-stage diabetic retinopathy, and there's not much left they can do for you other than tell you when you qualify for disability. You see, over the years, chronic high blood sugar leads to a lot of damage within the eye, namely inflammation of the retina and destruction of the blood vessels. At a certain point, the body tries to compensate by increasing this protein VEGF, which makes new blood vessels within the eye, but these blood vessels aren't very good at their job. They're very leaky, and they cause even more damage to occur, ultimately leading to permanent vision loss. Unfortunately, even though most of the damage occurs in the earlier stages, there aren't very many symptoms, so patients don't even realize that they have this condition if they're not getting their annual eye exams until much later. Most of the current therapies on the market don't address the disease until this later stage, and they only work efficiently in about 50% of patients. So what if there was a way that we could address this damage before it begins? That's where our product PhotoViz comes in. This is a novel microRNA inhibitor that helps to protect blood vessels and slow the progression of the disease before most of the damage occurs.
So now we know the problem, but the next question is, who is affected by diabetic retinopathy? Well, there are 643 million adults globally by 2030 that will have diabetes. Of that, 22.3% will develop diabetic retinopathy, and that equates to 143.4 million people that are at risk of vision loss. And the next thing you need to know is that diabetic retinopathy is the #1 cause of preventable blindness, and the key word is preventable.
So we've mentioned that there are drugs on the market that are meant for diabetic retinopathy. So where does Photovis land in comparison to these other therapeutics? First and foremost, Photovis is significantly more affordable than any of the other options. In addition to that, it targets multiple pathways. So it can be utilized in the early stages as a preventative, as well as the late stages to hopefully reverse the effects that are seen. In addition to this, it's disease modifying. So rather than putting a Band-Aid on what is already happening, you're fixing it at the actual source of the problem. And finally, it has the potential for repeat treatment, but our hope and goal is to continue to process it so that way it's less repeat treatments, which will be more financially affordable for our patients. So as previously mentioned, we'll have a $500 sell price, and that comes from a 93% gross margin with a $35 manufacturing cost per dose. As far as it goes for how many people are going to be seeing this, if we consider a 5% market penetration, we have a 32.2 million estimated market capture, which means that there's a lot of money in diabetic retinopathy. In fact, by 2034, we're expected to see an $18 billion market. And in addition to this, we do have some financials considering the insurance that it would necessarily be covered by using Medicare Part B reimbursement as an example.
So to discuss our timeline, our plan is structured in a way to get the FDA approval and get ready for a commercial launch in the US by 2034. We plan on beginning with preclinical IND enabling work, which should be completed by 2027. And in parallel, we'll initiate GMP manufacturing scale and enable the preparation for phase 1 clinical trials. And we will seek breakthrough therapy designation, which will potentially reduce our timeline about 2 years. Now, after that, in 2033, we will submit our NDA application, and with FDA review and approval anticipated within 8 to 12 months. And in 2034, we then expect a full US commercial launch, followed by post-marketing research in 2035.
So where are we getting money to fund this research? So our preclinical phases are completely funded, estimating to be costing us about $2.5 million. We have great financial partners with the National Eye Institute, Breakthrough T1D, as well as the SBIR STTR seed funding. In phase 1, we have the majority of it with our financial partners for the Foundation of Fighting Blindness, as well as the SBIR STTR, totaling to $5 million. So the last $2 million is where it comes from you for the advancement of vision care.
So as you know, we're asking for a $2 million early investment. This is because we plan on having a phase 2 acquisition in which our company would be valuated at $1.6 billion. Should this acquisition not occur in the year 2034 to 2035 of launch, we would plan to see a 10x ROI as well as the break-even point. And then if the acquisition were to occur, we would see 120x ROI. Our peak revenue would be seen in 2041 at approximately $8 billion. And then we do see a slow decrease. However, the revenue would still be in the billions. This decrease would only occur because the patent protection would end in 2041, and we would begin to see biosimilars hitting the market.
We do have some clinical partnerships. Specifically, we have here Molly Welsh, who is a visual electrophysiologist and certified orthoptist at UPMC Children's Hospital of Pittsburgh, as well as the Mercy Hospital Vision Institute. And she is quoted by saying, "Photovis represents a powerful and promising addition to the future toolkit for preventing and treating diabetic retinopathy, addressing the damage before it even begins." We would also like to take a moment to thank all of our consultants on the business as well as the scientific side of things. And we'd like to take a moment to thank you for your time and attention and ask, do you see the vision?
Mike.
So you told us that this inhibits some microRNAs, but you didn't really tell us much about mechanisms. How the heck does this thing work? What microRNAs, what does it do, and how does it affect the vasculature?
Yeah, so Photovis works by inhibiting microRNA LET7B, and this is known to be upregulated in diabetic retinopathy, and this inhibits normal PI3K signaling, which deals with cell survival and preventing apoptosis. It also is found to increase FOXO3A, which leads to increased autophagy, so more destruction of those blood vessels. So Photovis coming in is complementary to that LET7B, so it inhibits that and restores that normal signaling pathway.
Okay. And is this a one-time treatment or over a period of time?
In the future, we would like for it to be a one-time treatment, but obviously with RNA therapeutics, there are some considerations with stability, as well as, some considerations with stability, but we have some data that suggests that if it does have to be a repeat treatment, it would be less frequent than our competitors, so probably only 2 to 4 times per year, as opposed to.
So how would that compare to steroidal injections?
The anti-VEGFs is the major competitor that is most comparable to Photovis. So the anti-VEGFs occur once every 3 months at that.
Is that an intraocular injection into the vitreous? Yep. What about the damage of the mechanical penetration that happens with those sort of things.
Yeah, that's absolutely something that we want to further consider, and we won't be able to further consider that until we get through more of the preclinical and clinical trials.
Thanks.
If a patient stops being diabetic due to GLP-1 use, will their eyes continue to need this treatment, or do you see your addressable market shrinking as GLP-1 therapies increase?
That is a good question. A lot of so one of the biggest indicators is not necessarily mean blood sugar, but how long you've been living with diabetes. So that damage accumulates a lot over time. So they will still have damage within their eye. It's not going to go away overnight. So our product still would be able to help them by securing those blood vessels that are still damaged.
And as previously stated, we aren't just putting a Band-Aid on it like a lot of the anti-VEGFs are. That just stops the progression. We are fixing it at the source, which would essentially reverse the damage created. So even if they do no longer have diabetes because of the GLP-1s, we can still reverse the damage caused by the diabetes and restore their visual potential.
Maybe because of marketing, but it just seems like this is a highly competitive space. I know you had your competitive analysis there, but are you aware of like other things that are sort of in the trial stages or at the sort of a little bit further along than you guys that are sort of being studied right now?
There is one competitor that we know of that is currently in phase 2 clinical trials. However, it can't be used at the earlier stages like PhotoViz can. The one thing that it does have up on us is it is a hydrogel. So that's just a one-time thing and it consistently releases medication. But once again, it is not preventative like our product is. So we still have the edge over them.
Aya?
I think I am good, actually. Thank you.
Okay. All right. Well, if there are no further, oh, do we have another question?
No, I said we're all good, apparently.
Okay. Yeah. Okay. Well, yes, you must have satisfied every issue. So well done, team. And yeah, I've been wired up. Okay. Okay, thanks, Linda. All right. So again, it's another stellar year.
I'm going to use the mic so that people can hear you.
I'm mic'd up. Yeah. So another stellar year. Wonderful, wonderful presentations, everyone. Just a reminder of what we're all competing for. First prize is $2,500.
I like the way you said that.
Yeah. Second place is $1,500 and third place is $1,000. But of course, on top of that, for anyone who pitched a drug, there'll be, and you place in any one of those 3 places, there will be a $300 bonus. So at this stage, we're going to leave the judges in this room so that they can confer. So I'd ask everybody else who isn't a judge to step outside. Is there a reception? Yeah, okay.
We like to judge under the
Feed the
influence,
judges.
yeah.
And of course, the prizes will be announced during the reception. Marty, can you hear me?
Yes.
Well, because you are a team mentor,
Right.
we're going to have to ask you to leave the judging session, if that's okay.
I'm leaving. Good to see you,
Yeah.
Rob. Thank you.
Okay.
All right. Bye.
I think I'm good.
I'm here.
Yeah, Brad is going to Brad's going to want you to rejoin the meeting. He's going to close it down as it currently exists, and then use the same link. Yeah, and then how much time do you need, Brad, between closing it? Okay, yeah. So if you just do on in another minute or so after
Okay,
Brad
so
closes
not
it.
join the Teams link that is telling me judges deliberate to decide that I have over here.
Yeah, just join. Yeah, just join this link.
Okay.